what sort of data?
Everyone, can you see my screen and can you hear me well?
Yes, thank you.
Okay, great. So I will talk about our supplement work that was funded by NIH Office of Director by this mechanism. It's about human AI loop in cloud for distributed computation. So a little bit of background of what we do. We look at renal tissue, like any pathology images, thin tissue pathology images. So here is an example. And this is a kidney needle core biopsy. Typically these images are like 30,000 times 30,000 pixels, pixel large. And one of the small locations, if you zoom up, you will zoom in, you will see that a structure, which is called glomerulus. Same way, if you zoom in, you will see other multiple structures. So we use machine learning tools to study very large renal tissue images. In our lab in particular, we also look at other type of tissue images, but right now we're funded mostly to look at renal tissue images to understand underlying microstructures, the role of the resulting features in disease trajectory prediction. So at the heart of our work, we have developed this tool called Distributed Human AI Loop. So this part, what we developed, it is funded by this Office of Director by Hades group. And the goal here was whatever computational analysis that we develop, can we bring that and make it available for the end user via cloud-based platform, which anyone like at the end user side or any stakeholder can use that without knowing too much about the computer processing, so that they can use this tool. And we are currently trying to understand like how what are the various aspects involved and how we can improve this kind of tool. So imagine that we have multiple centers that are generating data, possibly scanned medical image data or tissue image data. They either can come in a central repository or they can be scanned in their original locations. Once they are scanned, they gets to in a queue in our server, and once they gets to in a queue, it gets to the underlying neural network model for processing, mostly for segmentation and feature extraction. And once they get segmented, the tool that shows you the visualization of the structure or structural segmentation. But as we know that these histology images or histological staining of tissues, they go through several types of pre-analytic variations, including imaging, tissue preparation, tissue sectioning, staining, and so on and so forth. So when we deploy this tool, if we try to deploy this tool in another person's data, or someone is trying to use their data using our tool, they may not get as accurate segmentation as possible. And that's where this human interactability with the system comes into play. And it also allows a human to be in control of an AI system toward auditability of the system. So then human reviewers, they review it, and once they review it, then they change it or then they correct it based on their desired performance, and then we train the network again, and this process goes in an iterative way, and slowly the segmentation performance improves, and this is what we call a human AI loop. So this is our system, and it's available at athena.ccr.buffalo.edu. Anyone wants to use that, they can register via this website, and I will just demonstrate this system now. I don't know how much the time permit to go through all the different capability of the system, but I will demonstrate the system. So can you see the online portal here? Are you right?
Yes.
Okay. So this is where we have hosted this tool, and it is being developed in our program, the funded mechanism. We are collaborating with a company called Kitware. So I would specify what are our respective contributions. So what Kitware came up with, something called Digital Slide Archive. It's nothing but like a box platform or Dropbox platform, but in a smart way, where you can keep thousands of these large digital whole slide images, which are very large. In fact, 30,000 times 30,000 or 40, sometimes they can be 40,000 by 40,000 pixels, depending on how what tissue you are scanning. And it also has a smart metadata archival system available, which basically store the metadata of the data in a smart way. So so here is an example. So if I try to open an image here, here if I open this image, for example, this image. So if you can see that this is a renal tissue histology image, and this is available via web, via the cloud server, and this is a deployable Docker in a Docker container, so anyone can get this software and they can use this. And here is an image, and you can see the different structure, and you can see that. And so this is the visualization portal. So what were our contribution mainly, or really, is adding these plugin buttons for end users. And as you can see, this segmentation button, so here are various different operations are there. And we also have other operations which are going through. I mean, we are not going to talk about those, are for cell detection. I will not show the cell detection tool today, which can count all the different cells for a particular cell type. But right now I'm going to show segmentation of structures. So we have this segment WSI part available, where you have it comes with auto populated with what would with the numbers, what would be the best for this whole slide image. And only thing you have to specify is the input image, where you can either input one image or a series of images. So here we'll just start with one image. So this is one image, and then you have to give a model file, that is one model that you want to use. For example, here you want to segment glomerulus, but you can segment other structures as well. And I wanted to tell that this is running in our cloud server. So depending on the GPU resources, we can parallel, we can write the code in a parallel, processing manner to with respect to speed. Right now, I mean, which has one GPU, but with multiple GPU cluster, whatever you are going to see would improve. So when you hit the submit button here, you will see the job started. And once the job start, if you can see how the backend Python code is running, and it would run. It would take a little bit to segment the whole slide image. You can see that it started, and something is running here at the backend. So it will soon it will keep running, and while it runs, I will show you some other things. Okay, so this is running, and but while it runs, I will show you some other things. So something is appearing on the top of my screen, so I cannot move this. Okay, somehow I need to move. Okay, so here you go. So so few other features while it runs. When it finished running, so you see this is running like 415 out of. So it basically divide the image in 72 patches and process each of them. While it finished running, the glomerulus would appear here in the screen. But here I want to, while it runs, I will show you something else. So for example, this is a large image here. For example, so in this is another image, and here you can see this is a large image. So basically, once the segmentation is done, you would see something like this. You would see all these glomerulus pop up in the other window. Okay, so you would see like the glomerulus. We can segment other structures as well, but I'm just using glomerulus as an example. So one beauty of the system is we have incorporated this structured segmentation capability. So namely, because of the tissue variability, because of how pathologists operate, they may not agree with this structural boundary, and one is able to correct this boundary. We can improve this boundary correction procedure, but right now this is what we have deployed. And the whole idea here is if you can correct this, then you can retrain the system. And I will talk about that once other ones. So it is still running. oh, it's done. So possibly this would pop up. So you see this one is done running and it just popped up. Someone might argue that it is still slow, but remember this image is very large and we're using one GPU and we can use multiple GPU cluster to improve this. So again, I mean, you can correct these segmentations, and in a batch of image, once you correct the segmentation, the idea is like if you have a batch of 1,000 slides, you correct, like, qualitatively go through all of them, and there is this button would let you go through all the images, and once you correct the worst ones, you can retrain the model. So we also have a training plugin, like train network plugin available with train network plugin available. So that is segmentation. Yeah, this is the train network plugin. So it wants you to give a training data folder, output model name, type of structures that you want to segment, because we can segment multiple compartments. So training layer would have multiple names, either glomerulus or glomerulus and tubules, and that has a detailed user users manual that we have available in the main home page. So if you go here, so here we have this manual available, the documentations that could allow you to use this tool. So this is the PDF file where we have the detailed documentation. I will not go through that, but what I wanted to specify because of the time limitation. So here, the mouse image I was showing, I mean, the way we are developing this system, and we are still moving, is we are really trying to address this FAIR principle of NIS use of data. So, so one thing that that once you segment, we are able to quantify deep features from this image. Deep features means the way we see that how big this glomerulus is, how many nuclei are there, how much is the texture is there, or different type of features. And once we quantify those features are written in this metadata tag of this tool. So when we talk about this metadata, I mean, it can include the other information, like the subject information, what type of tissue is this, p-analytic variable, sex, ethnicity, and all the information. So the whole idea of this metadata panel is there, which is still we are improving, is. And Sophia, you have to tell me when I have to stop. Okay, I mean, I cannot see what's going on in the chat button.
Okay, you have 1 minute.
Okay, I will just finish very soon. Okay. So the whole idea is that so these goes hand in hand with the standards, and one can deploy ontology, a nice metadata ontology here. And the other thing is that we have a plot option, so metadata plot option. So if one wants to see different features, so you can glomerular area versus say PAS positive area, for this bunch of data they have, they can add a color, so PAS positive area, and then once you save, this would appear here, and then you can see this data. So each of these dot is a glomerulus, and depending on feature type, any feature or any metadata, you can see them. And the whole idea is to extend this tool to address the interoperability of the system. Imagine that a physician is looking at their slide, and this tool that we're developing, if we can make it globally, a global repository using this tool, someone with a suspected tissue, like where they are not sure of the lesion or the diagnosis type, they can calculate feature, and they can kind of project the data here, where they can query using this system to find out what kind of other disease types that kind of matching the tissue they're looking at. So with that being said, I will just show very quickly what are the different type of things that we can segment. So we can segment basically in kidney space, all the different compartments, all possible tubule, glomerulus, that is glomerular interstitial space. We have a tool for fibrosis segmentation, interstitial fibrosis and tubular atrophy. We can segment arteries and arterioles, which is very interesting to me, because these data are taken from GTEx database that NIH has put together. These are from different.
Dr. Sarder, obviously you have a tremendous amount of very exciting material to present here, and thank you so much for your presentation. I think there's a, I'm certainly very interested, very
No
impressed.
problem,
and
no
very
problem.
interested.
Yeah, we,
So
we
I'm
can
gonna,
keep going.
I'm-
Yes, yeah.
I'm
Yes,
gonna
please.
shift now. I think we'll have to take your questions in the Q&A, and we'll shift gears now. Thank you so much, Dr.
Sure.
Sarder.